Greetings to everyone, and this is Luca Dellafiora from the University of Parma, Italy, and I'm very happy for the opportunity to present again our work. And as you can see on the slide, the presentation you are about to see deals with the use of computational method to study the interaction between the spike protein of coronavirus and the human target receptor angiotensin 1 converting enzyme 2. And in particular, our work focused on the anthraquinone class of compounds as a possible model compounds to prevent the interaction between the viral protein and the host target receptors. So, just let me to make a premise to avoid any kind of misinterpretation and misunderstanding. I am a molecular biologist by training and a food chemist, let's say, by adoption. The core of my research is the analysis of the mechanisms of action underpinning the biological or toxicological activity of food-related compounds. So that's the right standpoint to read my presentation. I'm not a pharmacologist, I'm not a medicinal chemist, of course I'm not a physician. I'm a food chemist with a biological background trying to describe and to understand the mechanistic aspects of food-related compounds. So that's why we study Emodin and anthraquinone compounds. Emodin is relevant for food, mainly because it's very abundant in the plant kingdom. It has many biological and toxicological properties, and in addition, it is also produced by fungi and molds relevant to the food safety area, and indeed, it has been recently included in the group of the so-defined emerging mycotoxins. So it may pose a risk in terms of food safety. So that's why we study Emodin and anthraquinone compounds. So in our analysis portfolio, we have also defined target fishing approach. That's a hybrid computational experimental way to describe, to identify theoretically all the possible biological targets of a given molecule. And in the process of analysis of Emodin, the antiviral properties popped out. In particular, Emodin is able to prevent the interaction between the spike protein of the coronavirus discovered in 2000, the beginning of the 2000, and the human target receptor, that is the angiotensin 1 converting enzyme 2 enzyme, AC2. Such an inhibition may also result in a reduced capability of virus to infect cells. To be honest, at the very beginning, at the time, we didn't pay so much attention to this property, mainly because it's not directly related to the harmful effect of Emodin, of food containing Emodin mainly. But at the beginning of the 2020, with the onset of the new pandemic, we definitely started to work harder and harder on that specific property of Emodin, mainly because, as I said before, we are food chemists with the expertise to describe the mechanism of action. And we thought that describing the mechanism of Emodin's action in terms of antiviral properties was worth of analysis. And even if we could provide few information, the work was worth to be done to, let's say, make more clear complex puzzle, the complex scenario presented by the new coronavirus. So that was the story why we investigated the antiviral properties and of Emodin against coronavirus. So the story started from the evidence reported by Ho and co-worker that Emodin, but not only Emodin actually, was able to prevent in a concentration-dependent manner the interaction between the spike protein of the coronavirus and the human ACE2 receptor. And such an inhibition was also related to a reduced capability to infect host cells. So the mechanism was definitely relevant. The inhibition was definitely relevant, but there were no clues on the underpinning mechanism of action. And the main aim of our work was to describe a possible mechanism of action to explain such an inhibition. To do so, we first analyzed the protein-protein complex formation using the pseudo-fine computational alanine scanning, and that's a very cool way to estimate the energetical terms of interaction. I mean, we used that approach to identify the important regions to stabilize the viral and the human proteins complex. And what we saw was that the protein-protein contact surface, there were two main regions that were supposed to be, from an energetical point of view, extremely important for the complex stabilization. And you can see the regions in the slide on the right of the slide. Then we analyzed the surface of protein-protein complex, and what we saw was that exactly in between the two key regions we identified, there was a small pocket possibly able to host, to dock, to bind small ligands, such as Emodin and other anthraquinone-like compounds. So, assuming the capability of molecules to interact in that pocket, there were two possible outcomes. The first one is actually the possibility to prevent the interaction if the binding at that pocket is able to disrupt the protein-protein interaction. The second possibility is the promotion of complex stabilization if the ligand can bring additional stabilizing interactions. So what we did was the assessment of the capability of Emodin and the other few compounds tested so far for the inhibition properties, the capability of this compound to interact with the pocket, with the surface pocket. As you can see in the bottom left, I reported the experimental evidence collected by Ho and co-workers, and as you can see, Emodin and to a higher extent promazine were able to prevent the interaction in a concentration-dependent manner, while anthraquinone basically was not. It was supposed to be almost inactive. So, docking study mainly provide scores, computational scores that are proportional to the capability of ligands to satisfy the physicochemical properties of a given pocket. And it was extremely important to notice that the computational scores, the higher, and usually the higher the score, the higher let's say, the fit and the match within the pocket. Well, all the scores we collected were in agreement with the inhibitory potency reported so far. And that was, so it was, the capability of ligand to satisfy the pocket in terms of from a physicochemical point of view was linked to the capability to prevent interaction. So that was the very first data supporting the hypothesis that the interaction at that specific pocket could be the mechanism used by Emodin and promazine to prevent the interaction. Then we analyzed the match between the calculated binding pose and the physicochemical properties of the pocket, and what we saw was that actually anthraquinone was able to satisfy mainly the hydrophobic region of the pocket, while Emodin and to a higher extent promazine were both able to satisfy not only the hydrophobic region but also the hydrophilic region, and in particular the promazine using the amino branch chain was able to satisfy to the hydrophilic region to a higher extent compared to Emodin. So also in this case, we got an evidence in agreement with the experimental results supporting further the hypothesis of the relevance of such a pocket interaction, resulting eventually resulting in the prevention of protein-protein complex formation. In addition, we also checked the effect of ligands on the stability of the spike protein, and to do so we ran molecular dynamic simulations, and in particular we put the spike protein ligand complex in a box of water and we let it run for 50 nanoseconds, and we did that to check whether or not ligands were able to disrupt the geometrical organization, the, let's say, structural organization of the spike protein, because we assume that the specific organization reported by crystallographic studies is extremely important to have a functionally active protein. If a ligand can somehow disrupt such an organization, it could prevent the interaction with the host receptor, the cognate human receptors, target receptors. You can see in the plot, the higher the value, the higher the reorganization and the geometrical destabilization of the protein. As you can see, anthraquinone was almost not able to disrupt the starting conformation, while Emodin and promazine were both able to disrupt the starting organization. So basically, both the compounds found active in real experiments were also able to reorganize and to reduce the stability, geometrical stability of the spike protein. Looking at the regions mostly involved in such a reorganization, we saw that Emodin and promazine, but not anthraquinone, were both able to reorganize, to reduce the organization of one of the two key regions identified as important to stabilize the protein-protein complex. And that was a very, very important result to support the hypothesis we have in mind. We also looked at the interaction of ligands, and what we saw was that over time, both Emodin and promazine were able to persist at the surface pocket, at the protein surface region, the region close to the surface pocket, while anthraquinone was not, definitely was not, basically because it moved out in a one-way direction outside the surface pocket, as you can see in the slide. So we also analyzed the possible effects of Emodin on the spike protein AC2 protein complex. And just to make a long story short, what we saw was that also in that case, Emodin was able to reduce the geometrical stability of protein-protein complex. Also, in the case of complex, Emodin was able to reduce the stability of the spike protein. It was able to increase the radius of gyration that indicates the unfolding process, and it was also able to reduce the number of polar and non-polar contact, protein-protein contacts. So also in the case of the spike protein AC2 complex, Emodin was able to disrupt the protein-protein complex formation. So what we got in the end was a proposal of a possible mechanism of action to explain the capability of Emodin to prevent the interaction between the spike protein and the AC2 protein. So the next step was the analysis of the new coronavirus, of the spike protein of the new coronavirus. And in that direction, there are some data in the literature supporting the use of Emodin in a co-treatment to fight and to counteract the infection of the new coronavirus. And a degree of functional conservation between the old coronavirus spike protein and the spike protein of the new coronavirus could be useful to design new strategies based on Emodin. What we did was the comparison between the sequence of the old spike protein and the new spike protein. Let's say the spike protein of the old coronavirus and the spike protein of the new coronavirus, and we were not surprised to see how they are similar in terms of protein sequence, but also similar in terms of protein organization. Also, specifically in terms of the surface pocket, we ran the same analysis you saw for the spike protein of the old coronavirus. Also in the case of the new coronavirus, we saw the two key regions important for the protein-protein complex stabilization. We saw the same surface pocket, and we ran, of course, and molecular dynamic simulation. And what we saw was that Emodin was able to interact with the surface pocket to a higher extent compared to the old spike protein from a geometrical point of view. That means that if you look at the old spike protein and if you look at the new spike protein, Emodin should interact better within the new spike protein, the spike protein of the new coronavirus. So what we got in the end was the hypothesis that Emodin has a degree of functional conservation also against the new coronavirus. So just to recap, of course, Emodin is not and won't be the medication. We are not trying to sell that. And for many reasons, including the diverse toxicological properties of and activities of Emodin. But beyond that, the main important outcome of our work was the description, was the identification of a possible mechanism of action to explain the activity of Emodin. And such a mechanism of action could be used to design Emodin-based compound with a higher activity and a lower toxicity, but also to design completely unrelated molecules able to target and to bind such a region, preventing the interaction between the spike protein and the human target receptor. What about the fight against the new coronavirus? We really believe that this kind of approach can be very useful in such a challenge because it can be applied beyond the spike protein, for instance, to assess the existence, to check the existence of additional important virulence factors. It can be applied beyond AC2 to check the relevance and existence of additional targets of host cells, and it could be applied also extensively applied and systematically applied on a very wide array of molecules to identify lead compounds to develop effective and efficient antiviral drugs. And at the very end, in addition to provide, results in terms of identification of new violence factors, of new active molecules, or new target proteins, this kind of studies provides the mechanistic understanding of the phenomenon you are investigating. And we really believe that the understanding of mechanistic bases are mandatory to face this read challenge from a more informed perspective. And in that direction, computational analysis and three-dimension molecular modeling could be a really useful help.
At the very end, let me thank all the people that helped me in running the analysis and in doing this research. So thank to Professor Chiara Engineer Verna from the University of Parma, and thank to Dr. Jean Dudorn from the European Food Safety Attention, and of course thank you to you all for your attention. If you are interested to know more, to learn more about the story I just told you, you can find more details on the paper we just published on this topic, and you can find the link on the slide. And well, if you want to know more, feel free to check it out.